Hi microbe folks, this is spring 2020, March 13th. This is a series of lab videos since the campus has been closed due to the coronavirus pandemic. We've been working on chapter 10 in the lab manual, microbial growth media, and the last two videos were on selective and or differential media. And just a reference, folks, in your lab manual, the spring 2020 version, on page 111 in chapter 10, there's this table that describes, and sorry, you can't see that very well, that describes selective and differential media. It describes if there's any inhibitors present, what will be inhibited, what will be selective. If it's differential media, it will describe what is the basis of differentiating different groups of microbes. For example, is it hemolysis, or is it lactose fermentation, or maybe mannitol fermentation? And in the previous two videos, you guys, we did blood agar and enrichment in differential media. We just did McConkey's media, which is selective for gram-negative Enterobacteriaceae and is differential based on lactose fermentation. Folks, we're not going to do eosin methylene blue, but what we're going to do is we're going to flip over to page 112, and we're going to discuss our last selective differential media called mannitol salt agar. And mannitol salt agar is selective for salt-tolerant bacteria, so we're really trying to select for the genus Staphylococcus. And because there's different species of Staphylococcus, we want to be able to differentiate between them. So we're going to differentiate between the species of Staphylococcus based on their ability to ferment a sugar alcohol called mannitol. So folks, I don't have any mannitol plates with me, but again, we've got some great photos that Dr. Holland had downloaded that we used in lab. So folks, so with mannitol salt agar, the salt is sodium chloride. Most microbes don't grow well at high sodium chloride concentrations, but microbes like the members of the genus Staphylococcus that have evolved to live on human skin, which is salty from sweat, members of the genus Staphylococcus are pretty salt tolerant. So to inhibit the non-salt tolerant microbes, we use a sodium chloride concentration of 7.5% sodium chloride in our mannitol salt. And 7.5%, that's weight per volume, means we're going to add 7.5 grams of sodium chloride per 100 mils of medium. So, for example, on the lab exam, if I asked you how much sodium chloride would you add if you're going to make a liter, 1,000 mils of MSA, mannitol salt agar, how many grams of sodium chloride would you add? You'd say 75 grams per 1,000 mils, and that's going to give you 7.5% weight per volume of sodium chloride. All right, so that high sodium chloride will help inhibit most microbes. We're going to select for the growth of Staphylococcus. The most famous member of the genus Staphylococcus is Staphylococcus aureus, are really highly virulent, very aggressive tissue invader, often resistant to multiple antibiotics. But Staphylococcus aureus has a less virulent cousin, more of an opportunistic pathogen, and the less virulent cousin is called Staphylococcus epidermidis. But the less virulent cousin can also grow in high salt concentrations. So how could we distinguish between them? Well, the way we can distinguish between Staph aureus, which is growing on this side of the MSA plate, and its less aggressive cousin, Staph epidermidis, we're going to do it based on mannitol fermentation. So mannitol is a sugar alcohol. If the microbes can ferment it, they will make acids and the pH will drop. So what we do is we include the pH indicator phenol red in the mannitol salt agar. At acidic pH, phenol red will turn yellow. So if the microbe can ferment mannitol, the pH drops, the phenol red will turn yellow. So yellow is a sign that the organism is mannitol positive. It can ferment mannitol. If the microbe can't ferment mannitol, they'll have to start tearing apart proteins and amino acids as a source of carbon and energy. And when they rip apart the amino acids, they release the amino group as ammonia, and that's a weak base. So the pH is going to go up. And phenol red at alkaline pH is going to turn a red color. Sometimes they call it fuchsia. So the key here is knowing that Staphylococcus aureus can ferment mannitol, right? So growing on MSA, it's going to turn the medium yellow. Staphylococcus epidermidis cannot ferment mannitol. So when we grow it on MSA, we're going to have this nice red color indicating that no mannitol was fermented. So again, this is really helpful, especially if you have an abscess or maybe an infected surgical site. You take a sample of pus. Um, you would inoculate to blood agar and to mannitol salt agar. You're going to incubate, and if you see yellow colonies, yellow medium the next day, you are definitely going to be worried that that could be Staph aureus. If you have colonies growing but the medium remains red, you're maybe not quite as concerned because that's a less aggressive Staphylococcus epidermidis. Okay, we'll stop that here.